thankful to be here and to give this talk. I'm a cancer biologist and I'm responsible for the section of genomics and transcriptomics at the Fondazione Pisana per la Scienza here in Pisa. So let me start with this quote, which is from Leo Tolstoy's novel. And happy families are all alike. Every unhappy family is unhappy in its own way. I like to quote this because it represents exactly what we feel when we study cancer. So from where cancer starts? On the left, you can see that that's the normal chromosomal set of each and every human normal cells. So it's 23 pairs of chromosomes. And no matter what, that has to be it. It cannot change. On the right side of your slide, you have these unhappy families. This is the chromosomal set of cancer cells. So in the normal conditions, these cells would die because they are not compatible with life in a normal condition. But in cancer, the checkpoints are all gone. So these cells, maybe few would die, but usually their aim is to survive. And they do survive, acquiring all the potential they have that they exploit from their complete DNA and their genome, and acquiring also new characteristics that we don't know. So cancer possesses all the dimensions of complexity. It's made of different elements, and these elements are interdependent. So it's not a linear system. So that it's not 1 + 1 = 2. And they are connected, these elements, and cancers evolves and adapts to the environment. So there are two lines of evolution in cancer. One is the linear one, where basically you have this consequential accumulation of mutation, and there are clones that outcompete the preceding ones that are lacking the fitness advantage. Then there is the branch evolution model, where basically you have different clones. Maybe they start from the same ancestor, but all these clones have different molecular profile, have different response to a treatment. And this makes the tumor very heterogeneous. And this heterogeneity is what fuels resistance to treatment. So there are technologies and approaches that I will show to you, some innovative approaches that we are using to dissect this complex clonal architectures of cancer. So what is the pathology that we study? The pathology we study is glioblastoma. Glioblastoma falls, unfortunately, into the branch evolution model, and is the most aggressive among the brain cancer, has a 100 mortality rate. There's no way out. There's no survival. The average survival is very short, and we, let's say, like to call it an intelligent tumor because it's able to escape any kind of treatment. And another really dramatic thing about this pathology is that temozolomide is the only chemotherapy that has been given in the last 15 years. So more than 15, 18 years, because the therapeutic field is stuck. And so it's necessary, it's urgent to find new ways to combat this kind of tumor. So our goals, they are all aimed to precision medicine, and we want to uncover potential targeting opportunities against glioblastoma, which are still lacking. And we want to improve the clinical management of these patients. So what is the issue that we at the moment have been addressing for the recent, during the recent years? Why is there such a high rate of failure for the drugs that are reaching the clinics? One of the reasons is that they are not reliable preclinical models on which to test the various compounds. So it's necessary to create models that would be more reliable, that would be viable, that would be patient-derived, that would be more viable in vitro, that would respect the complexity of tumor, maintaining the microenvironment, maintaining the cytoarchitecture, and also a kind of test to assess the drug response or the combination of drugs that would be fast and non-invasive on the tumor, because in case of glioblastoma, there is not even enough time for the patient to wait for the right drug testing. So what we wanted to do, we wanted to add complexity to what were the standard systems, which were testing drugs on 2D cultures. 2D culture are layers of cells, cancer cells. They are all the same, so it's basically an aggregation of cells. And so we wanted instead to move to the 3 organoids, or explant, as we call them, models, where you actually have different elements all connected together. And as you know, all these elements are creating different layers of organization that they're all connected with each other, creating a complex system. So we move from a layer of a 2D layer to an organoid, a piece of tissue. And this is what we've been doing in our lab. So this is an example of a tumor tissue collected from the surgery room, and we dissociated in little pieces, and not up to the single level, because that's not what we want. So we keep these pieces together. They are not that big, but not even that small, and we grow them in culture in a matrigel. So this is an example of one of these pieces that is growing in vitro. So you can observe how it's growing. It's growing very fast overnight. This is a video taken overnight in few days from the culturing. And we can appreciate that it's alive. So what is the technique and the approach that we use to actually assess the drug response? So we started with imaging. I mean, I'm a strong believer in checking the images, observing the tumor, because I think that the imaging the tumor is taking a snapshot of the tumor when it's in vivo, when it's alive. As you can see here, these are not, of course, live tumors. These are sections of fixed tumors, so they are slices, but still represent what the tumor was when it was in vivo. So you can see on the left, we have a breast cancer. On the right, it's a glioblastoma. But you can see how cells, they have a purpose, they have a mission. They are not randomly moving everywhere. So on the left, you see this basal membrane, which is broken, and cells are invading the parenchyma on the other side. On the right side, you see cells that are distributing themselves like is called pseudopalisade cells, and that's a reaction they have when they are in a hypoxic region. So they are basically escaping. So all these movements, all these are behaviors that have a control. But what is the imaging that we are actually using? We are using FLIME metabolic imaging. Okay, why metabolism? Metabolism is at the basis of all the actions of a cell. And it's in the literature is well described how the changes in metabolism could be related to the response to a drug. So when a cell is responding to a drug, the metabolism changes to a lower, let's say, to an oxidative phosphorylation metabolism, so less proliferative. And by studying NADH and FAD, which are 2 enzymatic cofactors, you can detect these changes. We got inspired from the work of Melissa Scala in 2014, where she basically performed FLIME metabolic imaging, and it consists of, these are cofactors that are naturally fluorescent. So it's not invasive. You don't have to dye or use any kind of dyeing. So they are, if they are excited with their specific wavelength, they will have a specific decay. And this decay, it's related to their molecular status. If they are bound to a protein, they have a specific decay. If they are free, they have another one. If they are bound, it means that they are in a specific metabolic state. If they are free, they are in another one. So what we did, we simplified what she did because in view of using it in a clinical setting, because it was too complicated, the way she was using it. So we concentrated only on NADH, and then basically what we did, since these compounds have this bio-exponential model, so determining the lifetime could be challenging. So Gratton, this Italian physicist that lives in the States now since many years ago, he developed this approach, the phasor approach, to study these lifetimes in tumors, which are complex. And basically what it does is for each pixel of the image, the lifetime is transformed by the Fourier transformation, and the pixel is put into this semicircle, and on the semi-universal circle you have the pure forms of the compounds. The blue is the free form and the red is the bound form, and then all the pixels fall onto this line, and the relative distance from these points, it's giving you how much of that form is present in that point of the image, how much bound form and how much free form. So by doing this, we were able in glioblastoma to develop this approach. And we used TMZ, temozolomide, as a benchmark, but the approach is meant to use it for any kind of treatment. So as you can see in this image, on the top you have the bright field image of the organoids on these explant pieces of tissue. Then in the second row, you have the middle row, you have a typical film image where you have these lifetimes. And then on the bottom is an extrapolation of the data of the previous image by using phasor approach. So we were able to extrapolate distribution curves and comparing always with the control, so with the tumor that is not treated. If the distribution curves overlap, we classify it as a non-responder, otherwise as a responder. And of course, all this was then characterized, molecularly speaking, just to prove that our classification was okay. And we were able to classify our samples, but not only as responder and non-responder, but thanks to these images, we were able also to express the level of response, which is a very important concept in cancer, because if just one little part of the tumor is responding, it would be not efficient, the drug. And we were able to do this. We stratify our samples, and now we are basically doing this also for other drugs. And all the patients that are going under surgery in this area, then they have after informed consent, they have their tumor grown in our labs and tested for different kind of drugs. And we were able to stratify it and then do omics on it, and also identify some markers and some also new therapeutic markers. Then this is the second approach that we use, where metabolism is not used as a marker of response to a treatment, but is important because of its clinical relevance for therapeutic purposes and for tumor progression. The more heterogeneous, metabolically speaking, a tumor is, the more aggressive is the tumor. So we decided to use the same approach because we can do NAD film also on fixed tissue, not live. They can be fixed. These are sections of fixed tissue. And we applied the same procedure, looking for heterogeneity, looking for different state of metabolism inside the tumor and between tumors, of course. So in this case, you have the intensity. Basically, these images, gray images, are the photons released in each pixel, which are not influencing the decay. On the bottom are histological section images. So this is what comes out when you have the film image. So you will see all different areas. It's clear that there are different areas with different lifetimes, but it's not so messy. I mean, how it could be. There are areas with the same lifetimes. And if we can have a distribution curve of the different lifetimes all over the image, but we can also do the phasor map approach, which we, let's say, like it because you can interact with the image. If you see that in the middle, there is the phasor plot. There's this cloud. Those are basically all the pixels. With the cursor, we can select the pixel with the same lifetime, and we can see where they are located into the image. So it's clear that there are different, again, that there are different states, different metabolic states. So we are using this approach to study these images, but we are looking for better ways, of course, because the idea is to be able to analyze many different images that in a way that we could extrapolate the most informative data, the complexity, the heterogeneity, the patterns, the relation between the different states. And in this way, we could be able maybe to stratify samples or associate a specific heterogeneity to a specific clinical status. And then this is the last approach that we are using, which is, it's going a little bit backwards. I mean, first we're looking in the tumor as a bulk in a whole, even though we are looking inside it. But in this case, since we do not know how it works, the whole system, we decided to look at the specific elementary components of the tumor and how they interact with each other. Eventually, if this interaction can give us some understanding of the collective interaction behavior of the whole system. So not looking at the global structure, but at the local rules. And we got inspired by this work. That it's actually a book that I read written by Giorgio Parisi, where he basically established that these flights, this storm of starlings, they do not have a top control, a centralized control, but each element, each starling has organizes itself with all the other starling in the close proximity. And in this way, following specific rules, they will not bump into each other. And so they maintain all these wonderful structure and create this wonderful structure. So we were thinking, what about tumor cells? Do tumor cells behave following rules based on their intricate relationship? Can I look at the cells and see if they behave like starlings? And in some way, these interactions are related to what the tumor will be as a clinical status. As you can see here, this is a video of cells in a 2D layer of a patient, so derived from a patient, and it's clear that these are like thinking organisms as I look at them. They are not random, they touch each other, they have a mission. So what we want to understand is what is their language, what is their code, because they have a new language. And if we can decode this language, maybe we can know how to stop them. So what we are doing, we are tracking them. So with the tracking analysis, we are at the moment trying to understand their characteristic, like area, volume, dimension, and their trajectories. So as you can see, they can have a super diffusive trajectory, they have a confined trajectory, they can have a Brownian trajectory, so go random. But what we are trying to do, so these are just small pieces, I would say the first letters, because we would like to go further. But at the moment we are here. And so what we did, we took these values, we put like of 2 samples. We have done already like 15, 20 samples, but I'm showing what happens with 2, just to make it more clear. You see, in a 3-dimensional space, we put 3 values, which are area, trajectories, circularity, area. Now, I can't read, but the system already recognizes two kind of a group of cells, which are orange and green. But if we superimpose the two patients and the two tumors actually combines. And also you can see that maybe few cells are mixed, but in any case, this kind of watching at the tumor might be interesting, and we could associate it to the clinical status. So eventually we can even microdissect the cells. So go back to the cells, we can do some omics, we can see eventually if the ones that are moving very fast have a specific DNA or a specific transcriptome, and do analysis. So I'm done. So I thank you so much. And this is my email. Eventually, for me, would be very important. Eventually, if somebody is interested in finding and getting strong expertise in imaging analysis and in this complex system, because we want to go further and understand how also cells are interacting with each other, not only their shape, their dimension, their movement as single element, but how they interact with each other.
Thank you very much, Maria Chiara. Just a question is possible. We have a large.
Can I ask a question remotely?
2 questions. Okay, the discussion at coffee break. Two questions only.
Question. Thanks. A little question. In this context, how do you describe the angiogenesis phenomenon? Phenomenology.
No, this is a very good question. Of course, these are the limitations of a 3D model in vitro. It still has endothelial cells. We check for that. We do not grow these organoids for a long time because I didn't say this, but we do the measurement after 3 days in culture because we do not want the tumor to evolve too much so that it remains as close as possible to the tumor in vivo. So we check that the different cells are there. Of course, the blood and the angiogenesis for 2-3 days, it's not happening probably, or maybe it is, but endothelial cells are there, they remain there. Glioblastoma is characterized by these microvascular glomerular-like structure, and they are still there. We check that.
Okay, another question.
A very short comment. Well, I like very much when you quoted the swarm intelligence of Giorgio Parisi. And there are already groups that are working in that direction. And I have a paper that I would like to show to you. Because anyway, I would like to make a comment. Your statement that the intelligence of a cancer is like that of an orchestra, but an orchestra without director, there is no director. And in fact, I appreciate very much what you.
It's true.
What you told. There is no director.
No, there is no director.
Thank you very much for a wonderful talk.
Thank you.